Have you ever wondered how cells decide which genes should be active and which should stay silent? This process is called gene silencing. It allows cells to switch specific genes off, helping scientists understand what those genes actually do. Gene silencing is a natural process that reduces or completely stops the expression of a gene. When a gene is silenced, it cannot produce its protein, even though the DNA is still present inside the cell. Scientists intentionally silence genes for several reasons. It helps them discover the function of unknown genes, study how diseases develop, and design new treatments for genetic disorders, infections, and even cancer. When a gene is silenced, noticeable changes can occur. An organism's characteristics, or phenotype, may change, its response to the environment may be altered, and researchers can identify the hidden function of that specific gene. Gene silencing can happen at two different stages. The first is the transcriptional level, where the gene is prevented from making messenger RNA. The second is the post-transcriptional level, where the messenger RNA is destroyed after it has already been produced. Transcriptional gene silencing occurs directly at the DNA level. DNA methylation and histone modification make the DNA less accessible, preventing RNA polymerase from transcribing the gene into messenger RNA. When the promoter region of a gene is blocked, transcription cannot begin. As a result, less messenger RNA is produced, leading to reduced protein synthesis. In simple terms, blocking the promoter effectively switches the gene off. Histone modification reinforces gene silencing by adding chemical tags to histone proteins. These modifications tighten the chromatin structure, making DNA less accessible. Because the transcription machinery cannot reach the gene, it remains silenced. In post-transcriptional gene silencing, transcription occurs normally, but the messenger RNA is destroyed before it can be translated into a protein. Without messenger RNA, protein production cannot take place. One of the best-known mechanisms of post-transcriptional gene silencing is RNA interference, or RNAi. First, double-stranded RNA enters the cell. An enzyme called dicer cuts it into small interfering RNAs, or siRNAs. These siRNAs are loaded into the RISC complex, which uses them as guides to find matching messenger RNA molecules. The RISC complex then cleaves and destroys the target mRNA, preventing protein production and effectively silencing the gene. Gene silencing is an essential mechanism for controlling gene expression. It plays a major role in research, biotechnology, and modern medicine, helping scientists better understand genes and develop innovative therapies. Thanks for watching.